Okay, Pooja, the recording is also in process, so you'll be getting the updates. Okay, maybe after the hour when I complete the
Yes,
class, you'll get all the updates. Okay, beta. So
okay,
this is a
sir. Yes, sir.
continuation. Yeah, this is a continuation of our first lecture. So we are focused on
Mm-hmm.
why we have to go for this chapter and what we are going to focus on this chapter. So that is what I discussed in the first chapter when I'm giving you an introduction about this. That is all the organelles that we're going to discuss, or every part we're going to discuss, all are from a structural and functional unit of life that is called a cell. But the cell interns are made up of chemicals. In order to understand this chapter, you have to know the external structure along with the chemical structure, because they all are formed from the living chemical molecules. And cell is the only organism that exhibit metabolism, because the nucleoplasm and cytoplasm are the reaction center of a cell. And if you want to have a chemical reaction between the non-living thing, a chemical called screen is visible for you.
Yes, sir.
Yeah, what color I'm using?
In color.
No, what color pen I'm
We are
using
doing a
now?
spiral.
Okay, okay, you're able to see that. See, yeah. So what happens in this, that we have to create the optimal condition for a chemical reaction to occur. But in our body, we don't require anything to be added, because the chemical reaction occurs by themselves, because there is one biocatalyst, they are called as enzymes. And the enzymes will take up the role to complete their biological cycles. And you asked me that what is nucleoplasm and cytoplasm. So I have labeled here that the matrix of the nucleus is called nucleoplasm. The matrix of the entire cell before the nuclear membrane, or you can say between the nuclear membrane and the cell membrane, is called as the cytoplasm, where the chemical reaction takes place. Now, Pooja, what is your normal body temperature?
I guess 38.5.
Okay, 38. Okay, average it is 37 to 38. We'll keep it as 37.5, right? Or
Okay.
as per 38. But when you're getting some pathogen entered into your body, your temperature will be elevated, right? The temperature of your body will
Yes, sir.
rise, right? So when you go to a doctor, he may
Yes, sir.
have a thermometer, or with the help of just a physical touch, maybe on the head or in your cheek, you can easily, or in your neck, you can easily find out whether you have some symptoms of fever. Why do we have fever? In
Yes,
fever,
sir.
there is increase in your body temperature, right?
Yes, sir.
Why the microorganism are entering into our body? The microorganism have a route of entry, and our body is a very good medium for multiplication of microbes. So our body has
Mm-hmm.
all the essential requirement for a microorganism to survive. So we become the complete host for the microorganism. So when the temperature of the body gets elevated, that there is a change in your body temperature. This change is not acceptable to the microorganism. So in that given condition, microorganism cannot survive. So it is actually a defense mechanism which is called fever. So for this, we take some tablets. We should not take the tablets like paracetamol oftenly. Only in extreme condition we can go ahead with that. Are you able to understand better?
Yes, sir.
So when a pathogen encounters your body, it enters into the body. Temperature your body increases so that the microorganism that entered into your body cannot survive. Now we have to focus on the membrane, where we are focused on the lipids, and we are focused on the organization of a lipids. So I told you a lipid is made up of two important components. What are they, Pooja?
Fatty acid and glycerol, sir.
Yeah, a long chain fatty acids, and along with them they have a glycerol. So fatty acid and glycerols are there. This fatty acid and glycerol, they together form one lipid. These lipids are simple lipids, but our body is a complex
Yes, sir.
organism, and the chemicals are stored in a complex form. So for these complex form, what happens? A phosphorus molecules gets added up to a lipid, and they form an important component of a cell membrane called as phospholipid. Got it?
Yes, sir.
And I told you this is the fatty acid. These fatty acids are nothing but they hate water. So they are called as hydrophobic. That means they hate. Phobic means fear. Fear of water. So
Yes,
fatty
sir.
acids are not attracted towards water, but glycerol are hydrophilic and they are attracted towards the water. So a lipid molecules, when you take here, you have a tail and you have a head. The tail is completely water hating, but the head is completely water loving. The water hating property of a fatty acid is called as hydrophobic end. At the same time, the water loving end is called as hydrophilic end. And I told you how they are organized. Plasma membrane is made up of lipid and protein. These lipids are called as bilipid. Why bilipid? The two lipids are arranged in the form of layer. This is layer number 1. This is layer number 2, and the round what I have given you, it is actually a glycerol, and the green color which I given in a tail form, that is your fatty acids. So head is projected outside, tails which are hydrophobic and they are projected inside the cell membrane. This is how the organization of a cell is there. Up to this, we are focused with chemical composition. Any doubt in the previous class?
No, sir. Sir, now while explaining, I have the doubt. In plasma membrane, lipid will be in the form of bilipid layer. Such as bilipid layer is known is in that case, lipid is defined as simple molecules. As our body is a complex organism, then the lipid will get attached to the phosphorus to form phospholipid.
Yes. I'll tell you in property. This is called
Okay.
amphipathic nature of lipid. What is amphipathic nature? The lipids have two different
Sir,
ends. Yes,
head.
beta. Yes, beta.
No, I'm saying that because lipid is both has both hydrophilic and hydrophobic, it is known as amphiphilic
Yeah,
molecule.
amphi means dual. So amphi means dual. Pathic means the pathway. So two different pathways are there, and how the two layers are arranged. Your blood is actually an aqueous medium, right? If I give a fat
Yes, sir.
molecules, which end of the fat molecules will get attract towards water? Tail or the head?
The head.
Head will get attracted towards water. So when
Yes, sir.
outer layer gets attracted towards water, inner layer are water loving or water hating. So they have a water
Head.
resistant barriers are there in the inner membrane. Got it? Now,
Okay.
now we have discussed about the these are basic things that we have focused. What these are also basic things we have focused. So we are focused on the important constituents that upon a lipid layer. So RBC is the wonderful one. So in all the cell you can see that mitochondria or except mitochondria, other cells lipids are always the highest. Got it better?
Yes, sir. What is the hepatocyte and oligodendrocyte, sir?
Oh, you want to know about the what is
What is hepatocyte and
hepatic?
oligodendrocyte?
I'll tell you. Hepatocyte site means cell. Hepato means liver. So hepatocyte means what?
Liver cells.
Yeah, they are liver cell. Got it, beta?
Yes, sir.
Next is dendrocyte. You have a question with dendrocyte. What is dendron? Have you learned about some term called dendron
Ah,
somewhere?
nerves.
So what does this topic deals about?
Nerve cells.
Yes. So they are nothing but they are found in your neurological cells. Okay. Okay, beta.
Yes, sir.
Dendrocyte, neural cell. Neural cell. Oligo means few. Few neurological cells they have taken. There also the lipid concentration is very high. Okay, beta.
Yes, sir.
So I'll be discussing some more things now today. Proteins are there in that, asymmetrical nature of the arrangement of layers, heterogeneity, and transient continuity with the endomembrane system. So these are the following things, the four important property I have to deal with it. Now I told you that to a lipid molecule, if phosphorus is joined, what they are called as, you can see, it is a glycerol. To glycerol, we have a fatty acids, and with the alcohol is also arranged. So to this, what they've added one
A
phosphorus.
phosphorus.
So they form a phospholipid. If we have colon, what do you mean by
Yes.
colon? What
I
is
don't know, sir.
colon? Have you learned about
Chlorine?
acetylcholine?
Uh, I
No.
I don't remember, sir.
Acetylcholine. It is actually a neurotransmitter which helps in transmission of impulse. Okay,
Okay.
it's a chemical which is called as a neurotransmitter. They are called as
Okay.
neurotransmitter. So when I teach you about nerve physiology, I will be going with neurotransmitter. Okay, beta.
Okay, sir.
Okay, it is actually a water
Sir, I have
soluble.
a doubt.
Yes, ma.
Sir, in glycerophospholipid, glycerol, you said it's an alcohol, and we have fatty acid, and P stands for phosphorus. We are adding an extra alcohol, or they're talking about the alcohol present in glycerol, sir?
Alcohol present in glycerol.
Okay.
Okay.
Yes, sir.
Now we have phosphatidylcholine. What is phosphatidylcholine? It's commonly called PC. So it is actually an choline is actually an essential nutrient. It is actually a nutrient which is there in the neurological cells, especially on your nerve cell. Maybe the dendrocyte they can talk
Mm.
about. They are an essential nutrient only, a vitamin derivative, right? So they help in movement of some substances. I'll tell you what is that. Next, like this, we have lot
Mm.
of chemical like when amino acid called serine is added, sphingomyelin. Just remember this name. They won't ask you the importance. But when the topic comes about the
Mm.
communication, I'll tell you about phosphatidylinositol. I'll just deal about this phosphatidyl linositol. I'll
Mm.
deal with this. Okay, beta? Linositol.
Okay, sir.
Okay, now it is actually a type of phospholipid only, and it is a major component of a cell, and these are the various groups we have. The main thing of all these are to give you the mechanical support to the membrane, or they form a structural framework. So they give a unique shape to the membrane. So they form a structural framework of the plasma membrane, and they allow fast movement. Remember this one. They allow.
Okay.
See, all this I have searched some literatures and giving you. Okay, beta.
Okay, sir.
Yeah, they allow faster movement of. Later, you can send me your question paper pattern. Okay.
Okay, sir.
So they allow. They are actually structural framework. That means they give a mechanical support to the cell. That's it. Especially to the membrane part of a cell. That is the plasma membrane. Allows even not the water soluble. Water soluble can pass through. See the structure. Water soluble outer layer is exposed to water. So water
Yes,
loving
sir.
molecules can easily attract it. What about the water hitting end? Can they cross this end? See, these ends are completely
No,
not
sir.
easy to move. So to
Yeah.
hydrophilic molecule, that is water loving molecule, can move into the cell very easily and go out of the cell very easily because the outer area is
Mm.
exposed, but the inner area is hydrophobic. So what about the water hating like fat soluble substances? The plasma membrane along with this lipid, maybe
Mm.
they can add one phosphorus. This phosphorus will give a
Mm.
framework to transport the molecule in and out of the cell. Got it?
Yeah. Yes, sir.
Now, general organization of a
Sir,
member.
sir, can you go? Sir, I have a doubt. Can you? What is that? Sphingomyelin. Myelin I studied in. Myelin. It's like the protective. Like it's a protective sheet of nerve cell.
Especially on
What
the
is that?
axon, you can see that, yes.
Spino. Spinosin.
Sphingomyelin. You're asking about sphingomyelin, right?
I'm asking about sigmoidin also and about things. The spinosin.
So sphingomyelin and
Sir, in sphingomyelin we have a structure, that pink color structure, sir.
oh, you want to know what is sphingosyn? You want to know?
Yes, sir.
Okay, no problem. I'll tell you. So, what is sphingomyelin? Sphingomyelin is actually there in mammals. It is actually mostly in mammals. Mammals means like as we are also a mammal. Even dog, cat are also a mammal. In the mostly in mammals, you can see sphingomyelin, right? Do you know what is apoptosis? Have you come across a term called
No, sir.
apoptosis? Uh, Endocytosis, at least.
I've heard it
Apoptosis.
somewhere, sir.
Yes, beta.
I've heard it somewhere.
Okay, one more topic. Apoptosis is nothing but programmed cell death. Your cell has a 24 hours life cycle, right?
Yes, sir.
So ova have 48 hours, sperm may have 72 hours. So the life of a cell is determined by sphingomyelin. It is actually a type of components of a phospholipid. It's a cholesterol containing compound. So they help in programmed cell death.
Okay.
Endocytosis. Endo means inside. Cytosis, some in engulfing movement. I'll just use a term called engulfing movement. Don't worry, I'll be teaching you about endocytosis and the functioning of plasma membrane. Okay,
Okay.
that time you can ask me about this what is this about endocytosis. So sphingomyelin is actually
Yes,
a
sir.
type of protein joined with the, it's a type of proteinous molecule only will help in transport. Mostly if you look about the chemical components of them, the chemical components of them is actually a myelin. In myelin, one spingo, one chemical is added in that. Okay.
Yes, sir.
If you want to know about the
So,
full name of this, the full name of this is N acyl. When acyl group is added, sphingosine phosphoryl choline. Okay, I'll tell you now. It's a sphingosine.
so, uh, uh,
Yes, beta.
I'm waiting for the explanation, sir.
No, no, I didn't get your point.
I'm waiting for the explanation, sir, for you to explain.
Yeah, yeah. So you are able to understand what is sphingomyelin. Sphingomyelin is a protein molecule joined with the plasma membrane to give you one structural framework, which is mostly found in mammals, which helps in cell death and as well as cell engulfing property. And this sphingomyelin has a phosphorus in the first position. This is the second position, and this is the third position. The first position, there is actually a phosphorus and a colon. So a term called phosphoryl choline is used. Are you able to know about the first position, phosphoryl choline?
Yes, sir.
Second question is, what is sphingosine? Sphingosine is actually a fatty alcohol. Sphingosine alone, when you take here as a term, I'll use here. Sphingosine, it is actually a fatty alcohol. It is a fatty alcohol. To fatty acid, when alcohol is added, what is the main thing is, of course, they also help in. Okay, you want to know the structure, or what do you want to know in this?
Structure, sir.
Okay, this has total number of carbon like this. They have 18 carbon. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. It is there are 18 molecules of fatty acids in this. To this, we are going to have one protein called choline. One protein is added. To this protein, one more phosphorus is added. Got it?
Yes, sir.
So what is this is actually it's a 18 carbon containing fatty acid. There are totally 18 carbon in them. So sphingosine is made up of one protein, fatty acid, alcohols along with the phosphorus. Sphingosine is actually a type
Yes, sir.
of a. Are you there with me?
The alcohol. Yes, sir.
Yeah. Sphingosine when you take here, in you like cancer biology, right?
Yes, sir.
Because I've seen your lecture when you're there in grade 8. Do
Eight. Yes, sir.
you know what is SPL insufficiency syndrome?
Sir.
No idea.
Idea, sir.
Okay. In our body, there's one gene called SPL gene. In our body, there is extra information. There's a gene called SPL gene. So we look about a SPL gene. The main role when you look here, they help in release of dopamine hormone. You know what is dopamine?
And, I guess it is the, they say it as the happy hormone.
Yes. To release the happy hormones, we require this.
Okay.
But this SPL gene is actually not there in this SPL gene is mostly there in plants. But in animals, when we add,
Okay.
they release in dopamine. In case of plants, they help in fruit, flower, and fruit development. They help in flower and fruit development. Okay, so a lot of things are there because it's too early to know about all these functions because all these are coming up by one I'm going to teach you, so nothing to worry about that. Okay, beta.
Okay, sir.
So these areas I know that you will be having lot of you'll be having lot of doubts in this chemical structure. Don't worry, just I've given you the introduction and
Okay.
organization. When I go in the upcoming topic, you'll be able to understand in depth. Okay, beta.
Yes, sir. Yes, sir.
Now cholesterol. So cholesterol is actually an important component of a thing. If you look about cholesterol alone, as per my knowledge, when I went through the literature in Science Direct, they gave us 25 to 30% of the total lipids are cholesterol. Among the lipids, the cholesterol alone, it's a sterol family. The cholesterol alone is able to have 25 to 32 percentage among lipids. So plasma membrane is made up of bilipid or
Lip.
biphospholipid. Among the lipids
Yeah.
whose concentration is very high means cholesterol concentration is very high. And the cholesterol why they require means, okay, rigidification of the membrane, that is nothing but stabilization. The plasma membrane is dynamic, right?
What is dynamic, sir?
Dynamic means always active.
Okay.
Dynamic
Yeah.
means always they are active. So they
Yeah.
have to be very sensitive and they're very active towards any response from the environment or internal or external environment. But to stabilize their function and to stabilize their structure, because when they work too much, wear and tear of the membrane may take place. So for the
Mm.
stabilization, we require cholesterol. Got it?
Mm. Sir.
Now, I'm going to teach you about the organization of a membrane respect to the proteins. Okay, beta?
Yes, sir.
So what I told you is actually a simple one, but what now I'm going to teach you is actually how the cells are organized. So be very careful with that. Now I'll use red color here. So what I'm drawing now, what is
Mm-hmm.
the structure?
Glycerol lipid, sir.
Yes, it's a lipid, a phosphorus containing lipid, right?
Said no, sir. Like lipids, like last class, you said that glycerol will be about and in between layers we have a fatty acid. Like you said, for in order for entrance of fat soluble, water insoluble molecules. So mostly, the lipid will be in bilipid layer, and also it has the attachment of phosphorus.
Exactly.
Okay.
I'll let me use the same color for the fatty acid chain also.
Okay.
You know these structures you're very much familiar with. Now I'm taking
Yes,
my
sir.
eraser. Yeah, I'm taking one of my eraser. I'm just amputating these proteins here. I just amputated few proteins here. So we have got your glycerol. Why there is actually a left a gap? Let me take my green color now. I'm going to draw one protein here. So plasma membrane is made up of lipids and protein. So you know the
Yes,
basic
sir.
structure, basic
Proteins.
labeling. Let us take here. These are the bilipid layer, right?
Sir.
What is the time there now?
Yeah. Right now it's 5-2, sir.
It's 5 o'clock.
Yes, sir.
You're enjoying your evening, right?
Yes, sir.
It's a bilipid. Now I can see one protein. This protein is there between the plasma membrane. That is between the lipid layer. You can see one protein. Can you see this?
Yes, sir.
This protein is actually called as channel protein or passage protein or tunnel protein. Lot of names are there for the same protein, okay? Or transmembrane protein. But all together, for all this, we're going to give a common name. It is there between the lipid layer, so we're going to call them as integral protein. What is the protein called as?
Integral protein.
Integral protein. So that is what we're going to have here. So these are some proteins are there, peripheral proteins are there, and integral proteins are there. So we have the membrane constitute. That is the membrane constitutes. Number one, we have two important things. One is the lipid layer, next is the membrane layer. So we have some membrane protein here. One is integral protein, which is also called as transmembrane protein, and it is actually partially or fully penetrated into the cell. That's how the membrane, which I discussed now with the diagram, they are organized. These are called as integral protein, and they are partially or fully embedded inside the bilipid layer. Got it?
Yes, sir. So you're saying that within the bilipid layer there will be presence of integral protein.
For example, do you have your home in Coimbatore now?
Yes, sir.
Okay. Okay. Who's there now?
Tenants, sir.
Okay. Okay. For example, if you are coming, okay, your tenant is not aware of Arun sir, right? If you suddenly come, okay, this is
Yes,
Pooja
sir.
home. I'm knocking the door or ringing the bell, and they're asking, who are you?
Yeah.
I'll say I'm Pooja's teacher. They have to make a call,
Yeah.
and when you agree and when they
Uh-huh.
know only, they can allow only selected person to enter. Trespassers cannot enter
Yes,
easily
sir.
into your home, right? That is the
Yeah.
main role of this passage protein. The name says passage, it's a root protein. Tunnel, big vehicle
Okay.
or small vehicle, it will be determined. Transmembrane or a transport protein, or a lot of names are there. These proteins allow
Okay.
the in and outward movement of molecules to enter here. We require some proteins that is called as channel protein or tunnel protein. Got it?
Okay, so like as we already know that plasma membrane is to separate the outside environment and from the cell. So in order for entrance of any molecules, they have to come in contact with the channel protein, and if it agrees, then it allows into the cell.
Yes. So mostly what
Okay.
molecules means, okay, we have ions, or what I'll tell you the names also of the molecules that can be crossing through these areas. So you're able to understand one protein, and this protein is strongly bounded between or flanked between the lipid layer. But another protein is also there.
Yes, sir.
These proteins
Okay.
are easily removable. They are only attached to the outside.
Okay.
So what you can see now, this is an another protein, and this protein is called as, it is attached outside, right?
Yes, sir.
It is called peripheral protein. So combinedly, I have a common name for all. This is attached outside, so they are called as extrinsic protein. Getting confused somewhere?
Sir.
Are you getting somewhere confused?
Sir, I'm understanding it.
So we have the protein which is attached to the outside is called extrinsic protein. The protein which are attached towards the inner side, which are not easily removable, they are called intrinsic protein. Now to this you can see a lot
Yes, sir.
of molecules like carbohydrates or a lot of other molecules, they may be bound to this. Lot of other chemical
Mm.
molecules, even they are bound to the lipids also. Even they are bound even here also. So these have a specific role. What are the specific role? I'll tell you one by one. So you can see here that the lipid layers, these are your, These are your bilipid layer. The phospholipid molecule, that is, the head is projected outside, the tail is projected inside. That is hydrophilic head of the glycerol and hydrophobic end of the tail. They are amphipathic
Mhm.
in nature, as the tail is protected completely from the water. So this will give a, they act as a water barrier. That means water molecules cannot pass in through the lipid layer. So where the water molecule can pass through? The water molecule can pass through the integral protein. Got it?
Yes, sir.
Now you just have a basic idea about the arrangement. So we have something here. These are called as some receptors. They are receptors which is made up of carbohydrate. Lot of things are there, and we have proteins. Lot of things are there. We'll discuss about all that one by one. Now, what is required? I'll teach you. We have integral protein, which are partially or completely embedded in the cell membrane, and we have a peripheral protein. Can you see in the bilipid layer how the protein molecules are joined? The transmembrane is also there. They are fully or they are
Sir.
partially or slightly or linked with another molecules, or maybe or linked with another protein. So a lot of attachments are there among the proteins. Got it?
Yes, sir. Sir, the gray part is the lipid bilayer. That pink, what is that pink part, sir? It's like
You're saying about this?
in yes, sir. And also, it's down. You can see like we have a green color, and also can see like a line, like the
Proteins.
carbon attachments. The carbon at
The COOH, right? Do you know the structure of amino acid?
yes, yes, sir.
So they have a alpha carbon atom.
We
You have a
and
COOH.
hydrogen,
So you have
so
COH.
and an amino group.
Amino group is not shown here, but you can see the protein are bound here and ending with the amino group and carboxylic acid end. So we have an amino group. This
Okay.
R is a variable group. This R is represented here.
Mm.
Can you see this R is actually made up of phosphorus? I think this may be any other part apart from phosphorus. Blue color may be anything apart from phosphorus. There are some other chemical molecule and the name is not linked or signified in this given diagram. Okay, it's nothing but other
So
than
the pink, sir.
this one you're saying
It's
about.
attached to the phos. Ah,
Oh, you
it's
want
like
to?
the
Oh,
connection.
yeah. This one is a peptide bond. You know what is
Okay,
it? You know what is peptide bond?
like the bond between the amino acids.
Yes, that bond is shown here. Got it?
Yes, sir.
Not only
Yes,
this
sir.
bond, even if you look about this diagram, even it can be a glycerol also. It can be a bond or it can be a glycerol. It can be anything else. But they are saying they're attached to the membrane. It
Okay,
can
sir.
be a glycerol, it can be a glycerol, or it can be an protein, it can be anything else. But the name is not signified in the given diagram. Okay. So we have a integral protein and we have a peripheral protein. So now we are going to focus on the peripheral proteins and how they are arranged. You can see here.
Sir, sir, I
They,
have a doubt in the in integral protein, sir. Like you said, no, sir. Like for we have the presence of phospholipids because of the entrance of fat soluble molecules, because we have the tail in that because the tail is hydrophobic. The water can't pass.
mm,
So after the entrance of water insoluble molecules through phospholipid, then they come in contact with integral proteins.
yes.
Okay.
Not only that, we have lot of routes of entry. So I'll teach you about the routes of entry also. Okay, nothing to
Okay.
worry. Okay, beta.
Okay.
Now listen here. I'll just draw a
Yes, sir.
simplified diagram here. Okay, I'll use red color here. Just a single line is enough to show about the membrane. First, we'll learn about integral protein, then we can go for the extrinsic or peripheral protein. Okay, beta? Okay, ma'am?
Yes, sir.
Now, there are some ions. Say, for example, I can say Na plus. Now, I have an ion which is Na plus. I'll have some other, I'll just copy down so that I'll be having a lot of repetition on the same diagram. See, I have got here a set of protein arrangements. So I have the plasma membrane. I've given some arrangements for you to understand. Though we have got some arrangement, the question here is how they are completely organized. For example, you know what is Nokia? I don't know whether you remember this. Active transport and passive transport.
I don't remember, sir.
Okay. There's a role called as active transport where the molecules goes from the region of low concentration to higher concentration by utilizing some energy. So for this, we have three Na plus molecule. They have to go outside the cell. Then we have 2K plus molecule. They have to come inside the cell by utilizing energy in the form of ATP. This process is called as active transport. That means utilizing some energy, the molecule has to be transported. So what happens here?
Trans Sir, transport of molecule from lower concentration to higher concentration.
Yes, by utilizing energy.
Okay.
So what happened here? The Na molecules have to get inside.
Sir, sorry, sir. Can you explain Nokia alone again, sir?
Which one? In Nokia is actually
Nokia.
a mobile phone. With this, a shortcut is there to remember about how the
Uh,
molecules are able to move. That is inside and outside, how the molecules are able to know. So, in order to utilize the energy called ATP, we require from outside the cell. This is outside the cell. This is inside the cell. What happens here? The three Na plus molecules. Sorry. So we have three Na plus molecule. The three Na plus molecule has to go outside. So they cannot go through the lipid layer. They cannot go. They can grow only through the channel protein or the integral protein which I discussed with you. Only with the help of these protein they can move. Otherwise they cannot move. You got it?
sir.
Now at the same time, these molecules even they have to enter in. If three Na plus goes outside, how many K plus have to enter in? Two, beta,
2K.
yeah, two K plus molecule should enter in. For this
Why it
process,
has to be 2, sir?
it's the universal rules. Then
Okay.
only ATP can be generated to have a difference in the distribution of ions. That's called membrane potential. You'll be learning a little bit later. You could have learned, but you could have forgotten. It's about a membrane potential concept. Okay.
Sir, I not study that, sir.
Right. Ninth படிச்சிருப்படா இல்லையா?
Sir.
So no problem. I'll be able to help you. Now imagine I have a ball. If one is going like this, other is going like this, the ball will rotate in this direction, right? Say sir no.
Yes.
See Veda, I have a ball. See, I have a nice ball
Yeah.
here. What happens here? One molecules are going like this. Other molecules are coming like this. Because of this,
Mm-hmm.
this protein will have a movement. They will move like this, right?
Yes, sir.
One rolling movement will be created. At the same time, if I have a ball and one is coming like this, other is going like this, then this
Mhm.
ball will rotate in this direction. Now tell me in which
Yeah.
direction does this protein will rotate?
Protein will rotate the first direction.
They rotate in this direction, right?
Yes, sir.
During this movement, okay, during this movement,
Mm-hmm.
what happens? ATP is utilized. ATP is completely utilized. That is, it is an energy driven process, right? So during this process,
Yeah.
to this for this process to transport 3 Na plus molecule outside and 2 K plus molecule
Mm-hmm.
inside, I require some energy.
Mm-hmm.
The energy is given by ATP. Do you know ATP cycle? No.
Remember it clearly, sir.
Okay, we have one we have one nitrogenous basis. It's called adenine. Got it?
Yes,
The
sir.
adenine, if I add one inorganic phosphate, I'm just putting as IP, inorganic phosphate, they will form adenine. I'm going to call them as adenosine now. I'll tell you the reason behind all these things. Adenosine. To it adenine means what phosphorus add பண்ணா அது பேர் adenosine வாங்க. So adenosine கூட ஒரு inorganic phosphate add பண்ணாச்சுனாக்கா it becomes adenosine monophosphate. To adenosine monophosphate, if I add one another molecule of inorganic phosphate, they will form adenosine diphosphate. The same adenosine diphosphate, if I add another molecule of inorganic phosphate, they will form adenosine triphosphate. This is how the ATP are generated in the same
Yes,
direction. This
sir.
is okay. This is the way ATP are formed, where energy is stored. This is the way energy is released. So it's the opposite pathway. So here ATP is broken down into adenosine diphosphate with inorganic phosphate is used for energy. So for active process and for transport of ions, we require these proteins. If these proteins are not there, the molecules cannot pass through. Got it?
Yes, sir.
So there are some cases that one molecules can pass through. There are some cases that two molecules can pass through in the same direction, or in some cases that the two molecules will pass in two different directions. This type of transport by the plasma membrane is called as uniport transport of molecule. They can allow only entry of molecule, not the exit of molecules. Here, two different molecules can enter simultaneously. So this is called as symport. Two molecules can enter in the same way. Here they're going in opposite way. It is called as antiport. It may be
Sir,
uniport.
like,
Yes, 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 ma'am.
Sir, like the first structure and for the fourth structure, sir. Now, as there is an entrance of ions and exit of ions, in that case, like you said, the ball example. In that case, ATP will be converted, sir. But for uniport and symport, how will be the ATP conversion, sir?
For this, I'll teach you little bit topics are there. If I teach you, you'll be able to understand. Okay,
Okay.
I'll teach you
Okay.
about that. So we have a trans membrane, and we
Okay.
have how the membranes are linked. We are focused. Now you can see the transport channels are there, na beta.
Mm.
They will allow the entry or exit of molecules. Are you able to understand? That's why I gave you this diagram.
Yes, sir. So
Yes, 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 please.
that structure will be present within the within the integral proteins,
Yes, I'm
the
talking. Yeah,
channels
yeah.
that you
Till now,
showed.
I didn't went through the peripheral protein. I'm discussing only about the integral proteins. And
Yes, sir.
what I'm discussing about the integral protein, which are called channel proteins, which tells us about how the molecules are able to move in and out of the cell. Okay.
Okay, sir.
So if one molecule are able to enter, it is uniport. If two molecules are able to enter simultaneously, it is symport. If they're going in opposite mass, it's called antiport. Now the question is, you ask where the ATP. If it is active transport, only we require ATP. If uniport, symport, or even it is antiport also, if it is passive transport, they don't require any ATP. A simple diagram to understand this. Okay, red color for membrane, right? We'll use the same line forever. Let me take some molecules here. Green color, maybe not green, blue, maybe the molecules. So here we have low concentration of molecule. Here we have high concentration of molecules. If molecules are moving from lower concentration to higher concentration, for this process we require energy in the form of ATP. Am I correct?
Sir.
This process is called as active transport, which is always an uphill movement. They will go against the concentration gradient. These molecules are able to grow or able to go against the concentration gradient. At the same time,
What
if
is
these
concentration gradient, sir?
concentration of molecules, here low concentration of
Okay.
molecules, higher concentration of molecules, the molecules are able to move
Okay.
from lower concentration to higher concentration, right?
Yes, sir.
At the same time, if the molecules are able to go from higher concentration to region of lower concentration, so there is no ATP utilization. There's no ATP utilization. This is called as downhill movement. They are coming down. The molecules are coming down, downhill movement without any expenditure of energy. So this is called as passive transport. So if you ask me about whether ATP is utilized or not, I can tell you that it is not about ATP can be utilized or not. It's all about from where to where the molecules are able to move, whether from the higher concentration to lower concentration or from lower concentration to higher concentration. Got it?
Yes, sir.
Now, okay, taught you about transport. Only this alone, better only one diagram, but you can see that how many explanations I have given you. Can you please check here?
Yes, sir.
So, a lot of new things you are learning.
Yes, sir.
Now we have, if it's too burden, you just tell me, okay? I'll try to cut short it also.
Okay, sir. No, sir.
We have some anchors, receptors, enzymes. So this we can see in the upcoming classes: anchors, receptors,
Okay,
and
sir.
enzymes. So
Okay,
anchors,
sir.
I have to discuss something about your integral protein. Receptors, I must discuss about integral as well as the peripheral protein. Both I need to discuss. I need to discuss about
Okay.
integral as well as peripheral for receptors. And because I told you, receptors mostly are there in your extrinsic protein. Even intrinsic also has some
Yes,
of
sir.
them, but mostly we have some functions of that. That's why we have to focus on receptors and enzymes in the upcoming classes. Then one will go for the
Sir, I
things.
have a doubt.
Yes, yes.
I have a doubt. Like in peripheral, in intrinsic proteins, sir, that you said you have entrance of fat soluble molecules. Like mostly, it will be like the biomolecules, carbohydrates, protein, amino acids. Uh, uh, what are the molecules that will be able to pass? I'm asking, sir.
Okay. You want to know that? I'll use a term called
Yes, sir.
selective transport. Not all molecules can pass through. Only selected molecules can
Mm,
pass through based on the need of a cell and what type of cell it is.
okay.
Only, that's what I told you, I'm coming to your home. Only selected people can be allowed to enter into your home. Unknown trespassers cannot be allowed inside the tunnel or passageway. Only known person can be
Okay.
selectively adopted. Got it?
Okay, okay, sir. So as now I understood, sir. So the molecules that are required and needed by the cell can only be allowed to be entered.
Yeah, exactly.
Okay.
Got it?
Sir, and I also have a doubt. Yes, sir. Sir,